Okay, good morning everybody. InteRNA is a Dutch-based biotech VC backed by Aglaia Oncology Fund and InCav Capital. And I'm pleased to present you introduction/update on our progress to the clinic. And from the title you may have seen that we have a focus on oncology. Our lead candidate has a unique dual mode of action, which we consider as a 1-2 punch in IO. Has activity against tumor cells directly, but also is able to modulate the tumor microenvironment and thereby initiating a long-term T-cell-based immune response. As you heard from the previous speaker, Rna Therapeutics, so our lead candidate is a double-stranded microRNA, chemically modified and formulated in a third-generation lipid nanoparticle. And with that third-generation lipid nanoparticle, basically we challenged we solved the drug delivery challenge for our applications, that we can deliver our cargo beyond delivered to tumors elsewhere in the body. The underlying platform is based on a lentiviral-based microRNA expression library that allowed us to do multiple high-throughput functional screenings in cell-based assays with the hallmarks of cancers as phenotypic readout. And around this platform, we built several layers of IP claiming function on microRNAs with a patent life up to 2031 and additional filings on composition of matter that gives us patent life up to 2039. And that all executed with a team with complementary skills and knows how to bring a drug into the clinic in the oncology field. As I mentioned, we are focusing on microRNA therapeutics, and microRNAs are actually small RNAs transcribed from your non-coding DNA in your body and are the natural regulation mechanism to transfer your genetic code into functional proteins. They bind to messenger RNAs and thereby induce cleavage of the messenger RNA or inhibit translation to protein. But they can regulate more than one messenger RNA at the same time. You have 2,000 microRNAs in your body to regulate 20,000 genes. And these messenger RNAs are actually across the different signaling pathways and connected in networks. And thereby they have a different MOA compared to the current single-target drug modalities that you have today. But with the pleiotropic effect of a microRNA, basically we potentially have a longer-term control of the disease and reduced relapse rates of the tumor. And coming to the problem in the oncology field is that it's generally accepted today that hitting only one target, you do not have the home run in terms of efficacy for the patient. Tumor cells have the capability to activate resistant pathways. Tumor cells have the capability to produce decoy signals so that they can evade from the immune system. And with the anti-PD-1, PD-L1 checkpoint inhibitors in the market, they are here to stay, but they certainly do not represent the magic bullet in a lot of indications beyond melanoma and lung cancer. So with the pleiotropic multispecific messenger RNA regulation of microRNAs, we may have a tool at hand that can be a solution for the failing combinatorial PD-1 strategies. So regulating across pathways, making it much more difficult for the tumor cell to initiate resistant mechanisms, and next to that, also regulate pathways that are involved in the evasion from the immune system and pathways that are involved in the activation of the immune system. So our lead candidate, INT1B3, provides all this with the direct effect on the tumor cells and the tumor microenvironment modulation to initiate the T-cell immune response. One step back, so the company actually started off with deep sequencing and bioinformatics to identify novel microRNAs that at that time were not found by mainly Tom Tuchel. We cloned a selection of these novel microRNAs and known microRNAs in this lentivector system to allow for these high-throughput screens. And basically this platform yielded then in the preclinical pipeline that we have today on the right, with a focus on INT1B3, but we have other compounds lined up shortly after. And then we have also a CNS program in place to show that our platform actually also works outside oncology and basically for any indication. In the CNS program, we have two proof concepts, one for axon regeneration after optic nerve injury, and the second one is inhibition of spontaneous seizures in a model for epilepsy, and we basically are trying to partner that program for non-dilutive funding. So I'll give you some insights on INT1B3, which is basically a combination regimen in one drug. I'm going to show you some data on the direct effects on tumor cells, on the tumor microenvironment modulation, and then basically the observations that we have less recurrent primary tumor growth, we have less metastasis development and improved survival over anti-PD-1, and all are supported by this long-term T-cell response. Here you see an overview of the effects that we see in panels of tumor cell lines, which we don't see in normal counterpart cells, and that we also see in vivo tumor mouse models in nude xenografts, so mice without an immune system, like inhibition of cell proliferation, cell cycle arrest, and, for example, induction of apoptosis through caspase activation and PARP cleavage. And since we found CD39 and CD73 as targets for this microRNA enzymes that are involved in the adenosine pathway, we switched to syngenic mouse models to investigate the activity in tumor mouse models in mice with an immune system. And here's the proof of concept model for that, based on the triple-negative breast cancer cell line 41, where you implant the tumor cells in the mammary fat pad, and then we start treating twice a week, Monday, Thursday schedule at 10 mg/kg. And then we did effects analysis after the first week of treatment and the second week of treatment. And what we saw in the first week of treatment, we induce production of immune stimulatory cytokines like IL-2 and interferon that leads in the second week to recruitment of CD8 positive T cells, and nicely also downregulation of the immunosuppressive T regulatory cells. So bottom line is that we are changing the TF over Treg ratio from immunosuppressive being 1 to immunocompetent being 1.5. And we especially find this downregulation of Tregs an interesting feature, as you see that toll-like receptor 9 agonist, RIG-I, or CpG oligonucleotides can also make tumors from cold to hot by recruiting CD8 cells, but they cannot downregulate the immunosuppressive T regulatory cells. So this model, this 41 TNBC model, is a metastasis model. So after 2 weeks of treatment, you remove the primary tumor, and then you follow the mice for recurrence of primary tumor growth and metastasis development. And the first observation is shown here in 2 independent studies, that we have significant less recurrence of primary tumor growth in the INT1B3 treated group. And ultimately, this leads to reduced metastasis development and improved survival of these treated INT1B3 mice. And if you analyze the dead mice in the PBS and the anti-PD-1 group, you indeed see that the lungs are full with metastasis. So what we then did, we took the surviving INT1B3 mice and we re-challenged them a couple of weeks after end of treatment with, again, tumor cells, this time not in the mammary fat pad, but subcutaneously under the skin. And then we saw that these tumor cells do not form a new tumor anymore. So it looked like that these mice were protected against additional tumor cell challenges. And to investigate whether this was a B-cell antibody response or a T-cell mediated response, we took the surviving mice and divided them in 2 groups, with one group where we depleted the T cells using anti-CD4 and anti-CD8 antibodies. And then both groups were challenged again with tumor cells. And then you see that if you do not deplete the T cells, then these mice are protected against further tumor cell challenges. Whereas if you deplete the T cells, then you see normal tumor growth back up. And to investigate whether we could transfer this to naive mice, we did an adoptive T cell transfer where we took the CD3 plus T cells from the surviving mice and transplanted those in naive mice, so mice that do not see the drug at all, and actually the INT1B3 surviving mice haven't seen the drug for a long time. But if you transplant these immune cells and then do in these naive mice another tumor cell challenge, you see that also these naive mice are protected against further tumor growth of these injected tumor cells. If you zoom in with transcriptomic analysis on the biology, then we see mainly an activation of the tumor suppressive PTEN pathway counteracting the tumorigenic PI3 kinase and RAS-MAPK pathways. If you look at the individual messenger RNAs that are regulated directly by this microRNA, then you see the genes involved in the effects directly on the tumor cells. You see the regulation of the adenosine pathway, which is involved in the evasion of the immune system. So we decrease the transition of ATP into adenosine. Thereby, we see increased ATP levels in the tumor, which is already immune stimulatory. And then we have further regulation of new IO targets that are involved in the activation of the T-cell immune response. We then further embarked on a pilot tox and dose range finding studies to have initial insight in toxicity upon IV administration of this tumor suppressor microRNA. So we found for rat and monkey the dose ranges that we can use in our GLP tox later this year. And for us, giving a great comfort is that with these dose ranges, we are in the same ballpark as on Patro, which is the first approved product of Alnylam, which is a double-stranded siRNA formulated in the first generation LNP. Also notably is that Alnylam is actually dosing on Patro at a higher dose level in humans than was established with the NOELs in the GLP tox. Dose-limited tox going high, escalating dose levels in monkey. We see some AST, ALT elevations, but we do not see complement activation and no acute tox at all in monkeys. And we were able to establish the PK/PD relationship. So our draft target product profile is basically giving us sight on a first clinical trial with expansion cohorts 1B focusing on HCC patients which are sorafenib and PD-1 refractory, and also TNBC patients that are chemo-resistant and TNBC and anti-PD-1 refractory as well. So to enable a further clinical trial for this program, we are aiming to raise 25 million euros. That should give us a runway till the end of 2021 with a proof of concept in 1B in those two cohorts. This year focusing on finishing the GMP, CMC and the GLP tox. Next year the phase 1A pretty standard dose escalation study in all comers 3x3, and then the extension cohorts in 2021 and be phase 2 ready by the end of that year. The team that we have in place, I have a background in cancer research and also in corporate finance. Michel Génicot came in from J&J, where he oversaw the development of oncology drugs from pre-clin to early clin. And Lawrence came in from Profibrix, where he oversaw the whole CMC and regulatory from pre-clin all the way up to market approval, and then Profibrix got sold to the medicines company. And that's the story. I would like to leave it with that. Thank you very much.